Hi, this is Casey, and this is my cell division on mitosis. To begin, this is the G2 phase, the late interphase. This cell is in G2, which is the final part of the interphase. During this stage, DNA has already been replicated, and the cell is preparing to enter mitosis, and the chromatin is still uncondensed, the nuclear envelope is intact, and the centromeres have duplicated. So closer look. Then we go into early prophase, where the chromatin begins to condense into visible chromosomes, the spindle fibers start to form, and the centrosomes which begin moving toward opposite sides of the cell. The nuclear envelope is still present, which is that in blue. Also included the late prophase. By the late prophase, the chromosomes are fully condensed, the nuclear envelope has broken down. As you can see, there's no longer the blue nucleus. And the spindle fibers are now extended across the cell and begin attaching to chromosomes at their centromeres. Then we move on to metaphase. Here in metaphase, the chromosomes align on the equator, which is called the metaphase plate. Each chromosome is connected to spindle fibers from opposite poles. As you can see, they're lined up in the middle. We then enter anaphase, where the centromeres split and the spindle fibers pull the sister chromatids apart towards opposite poles of the cell. This ensures that each new cell will receive an identical set of chromosomes. And here, I wanted to explain that there's more in one cell than each due to space. That's why I just wanted to make it obvious of how those are being separated and formed. And if I used more, then yeah, sorry. And then here in telophase, during telophase, the separated chromosomes begin to uncoil back into chromatins. New nuclear envelopes form around each set of chromosomes, and the spindle fibers begin to break down. The cell is almost ready to divide. We then move into cytokinesis. Cytokinesis is the final step of cell division. The cytoplasm divides, and the cell membrane pinches inward to form two identical daughter cells. Each has its own nucleus and has its complete set of DNA, which is then where we move into G1, which is the early interphase, which could be the end of mitosis or the beginning of mitosis, depending on how you interpret it. After mitosis, the cell enters the G1 phase, the first part of interphase in the new cycle. During G1 phase, the daughter cells carry out normal cellular activities and prepare for the possibility of another round of division. My model shows the G1 phase as the beginning of the next cycle after cytokinesis. Hello again. Although my clay model clearly shows the stages of key mitosis, there are several inaccuracies that are important to point out. First, everything in my model is static. In real cells, structures like chromosomes and spindle fibers are constantly moving. For example, spindle fibers actively pull the sister chromatids apart during anaphase. But in my model, they're just fixed pieces of clay. Second, the model is two-dimensional and flat, while actual cells are three-dimensional. This limits how accurately I can show the spatial relationships between structures inside the cell. Third, the chromosomes in my model are already fully condensed in prophase. But in real cells, DNA starts out as the loose chromatin and gradually condenses. That gradual transition isn't represented in my model. Fourth, I used bright different clay colors or painted different parts of the clay to make each part of the cell easier to see and understand. However, in real cells, chromosomes and spindle fibers don't come in color-coded sets. This is just another simplification for the model. Fifth, the stages in my model are shown as distinct step-by-step images, but in real life, these phases can overlap. For instance, cytokinesis can begin during telophase. I separated to help show each phase more clearly, but it doesn't fully reflect how mitosis works in real time. Lastly, my model ends with G1 phase, which begins after cytokinesis. In G1, the cell grows carries out normal functions, and prepares to either continue through the cycle or pause in G0. While this stage is shown in my model, I didn't include all the organelles or functions that happen in this phase. So again, it's just a simplified version of what really goes on during mitosis. Thank you.